Hi, everyone. Hi. Welcome to this pitching session. I'm really happy. I'm Elodie Chabrol, and I'm really happy to have you today to hear all the pitches of the 15 PhD students and their artists that are going to pitch you in 90 seconds their projects with art. So be ready to listen to them. Please cheer them up on whatever channel you are, Twitter, LinkedIn, or YouTube. A very difficult challenge. They are going to show you the art. They are going to explain to you how they did it and everything under 90 seconds. So you're going to hear them very, very soon. Who are they? They are part of a international training network funded by Marie Sklodowska-Curie Actions. And they are all about enhanceropathy. So basically, enhancers are little parts of DNA that make the DNA less. And when it doesn't go right, it creates those diseases. So they are all working on that. You will hear about their work very, very soon. I'm going to start calling them. I'm really excited to hear them because I've trained them for days. So here we go. So the first, perfect, sorry. So the first one to join us are going to be Abhishek Agarwal and Tejaswini Padmas. So let me bring them on the floor. Hello.
Hello.
Hello,
Hi.
both of you. How are you?
I'm good. How are you?
Are you ready? You're good, good, good. Are you ready to pitch?
Yeah, sure. Why
Yes.
not?
Set.
Okay, perfect. So I'm disappearing.
So, hello everyone. My research project focuses on the studying of a unique patterns of DNA elements that regulates the gene expression, which are commonly called as enhancers and promoters. Additionally, several other proteins are also involved in helping these two to come together for the proper orientations of the genes. As a result, all of these components and the proteins are positioned on the DNA in such a way that they guarantee the regulate the expression of a gene. So now let's attempt to comprehend this using Tejaswini's illustration inspired by the natural world.
Imagine a lovely forest full of birds where a select handful gather and attempt to nest. They perch randomly on branches, sing, and are involved in other courtship activities to find a suitable mate. Similar to DNA, these they can seat randomly. The birds on the left denote enhancers, and the one on the right denotes a promoter. The berries above them denote the proteins that aid in the couple's union. This union results in the construction of a nest and laying of eggs, which is a sign of gene activation. This is just a sketch with a sneak peek of the final illustration, which you will see in the exhibition.
Wow, what a stunning and artistic representation of my research project by Tejaswini. I never thought that words can be used to explain my project. Can't wait to see the final product on the event which is in October in Marseille. Thank you.
Well done. Well done, and thank you for being the first one. It's always a bit stressful. It's really beautiful art, and as you said, yeah, that is the event in Marseille. We can't wait to follow you. During the pitching, you've probably seen in the YouTube comments all the links to follow both of you. So please, if you listen and if you're really interested and you want to know more, follow both of them. And I'm gonna thank you, and we're gonna switch to the next ones.
Thank
Thank
you.
you very
Bye.
much, both of you. See you soon.
Thank you. Bye.
And bye. And now we're gonna listen to Vartika Pish and Ivan Van Drager that I'm bringing on the floor. Vartika and Ivan. Yes. Hello.
Hello.
How are you?
Good, good. Very nice weather.
Ready to pitch?
Yeah. Mm-hmm.
Yeah, a bit too hot. Okay, so you have a pitch to the video, and you have 90 seconds to pitch. Removing myself, putting the video, and that's a go.
So, imagine a puzzle that has stumped doctors and scientists for years, a complex heart problem affecting newborns. In my project, I'm going to be tackling this problem head on. My mission is to become a genetic detective using advanced tools to unravel the mysteries within the genomes of these little ones. But here's a catch. Analyzing these genomes is like searching for a needle in a haystack. The amount of data we have to sit through is mind-boggling. It's like sorting through an immense mountain of information, hoping to find that one crucial mutation that truly matters.
Thanks. So my visualization represents an epigenetic landscape. It's created with textiles, dyes, shibori, and embroidery. And to me, these important mutations that Wartika is trying to find will be situated in a molecular landscape like this. So there's peaks and valleys, which mean a lot or little genetic activity, different genomic fabric at different sites, and numerous factors and threads. And Wartika has the big task to find out how things are connected at the molecular level and which thread might have unhinged all of it.
This is fascinating. The level of complexity in a single piece of cloth, it reminds me of the small details I tackle every day, tackling this very big problem. It's very surprising to be realized like this. I'm looking forward to the actual art now. Thank you so much.
Yeah, thanks a lot to you too, Wartika. From my side, it's been interesting and fun to work on this project with you. Thanks.
Hey, thank you. Well done. It's really nice. So we can work. Is it a project, or it feels like it's fabric somehow?
Yeah, I didn't fully get the question, but yes, it's all fabrics. So tulles and cottons, and then pressed into shape.
Ah, nice. Okay. Thank you. So we're gonna go to the next one. Thank you, both of you, for pitching.
Thank you.
Your links and everything are in the chat. So if we want to reach out to you, we can to get to know you more and to know more about what you do. So thank you very much. I'm gonna now say goodbye, and we're gonna welcome the next two ones. Perfect. So we're gonna welcome now Anna Setnarovska and Laura Holm. Wait, give me a second. Sorry, I'm adding everyone. Perfect. Hello, you're both together. It's nice to see you.
Yes.
So, are you ready to pitch?
Yes, we are.
Yeah.
Okay, let's hear you.
Okay, so the main goal of my project is to understand the specific mechanism, how and why the certain parts of DNA, which are called enhancers, influence decision-making of stem cells. So the undefined cells with multiple potential decide to become fat cell. And by better understanding of fat cell development process in the human body, we really aim to, and we hope to uncover new insight into obesity and other metabolic diseases.
So this is just a concept work for now, but I was really inspired by Anna deleting these enhancers in the genome. There are 9 squares on the picture. Each square represents a mutant cell line. And within each square are 9 pillars representing the enhancers that Anna is working with. And the short pillars then indicate the enhancers that she deletes. And the different colors kind of emphasize the different identities or lack of identity that the cells get when you alter or remove the DNA.
I really love the concept of how those pillars hold up the identity, and that you include those 9 studied sequence and how perturbation of them affect the the fat cell identity and modulate this identity. So that's the core of that project, to actually understand why those lines are so important for the fat cell formation. And I'm really looking forward to the final version. Thank you.
Thank you.
Amazing, well done. It's really, really nice, and thank you for putting energy in your pitch. It was nice. We look forward to following you and seeing the final thing. So again, all the links are in the chat. So if people want to reach out to you, they can do it. Thank you very much, both of you, for your pitch. We can't wait to see the rest later and see you on social media.
Yes, thank you.
It's time. You're welcome. Thanks. Now it's time to welcome Veronica Erconi and Dimple Devadasda is not there, but Veronica. Yes, hi Veronica.
Hi.
So you're teaching on your own, right?
Yes.
Okay. Let's hear from you. You have 90 seconds. Are you ready?
Yes.
Okay, perfect.
Thank you, Lodi, and hi, everyone. Imagine you are at a library and a book catches your attention. You open it and you start reading. You notice that some letters are highlighted in red. These are typos, but not all of them are really changing the meaning of what you're reading. Then you turn the page, you still see these red letters, but this time is much more difficult to understand which among them are changing the meaning because the language changed and you can't fully understand it. In my PhD life, this book is the genetic information of children affected by a rare type of diabetes, for which we still don't know which variant, that is a typo, is causal in that case. And this is crucial for us because knowing this, we could provide the most appropriate treatment to them. So my focus now is essentially to try my best to understand fully this new language in order to spot the causal variant for them. Unfortunately, Dimple is not here today, but I have to say that collaborating with her has been mind-blowing. It was crazy to see how a complex PhD project can be transformed into a piece of art that everyone can enjoy. I really love it, and I hope you like it as much as I do. I have to thank you all for your attention, and this is my speech.
Thank you. Well done. So yeah, it is amazing to see science transformed into art. It's really always a big surprise and always very, very nice. So Dimple isn't there today, but we put her links and yours as well in the chat. So if some people want to contact her, they can do it or contact you. Thank you very much, Veronica, for pitching today, and see you soon on social media or somewhere else.
Thank you. Bye.
Bye. So we're gonna go now to the next one. So it's a crazy rhythm because we have 15 projects to show you today. And now it's the turn of Anna-Karina Felder and Anneli Martens. So I'm gonna add them with me. Hello.
Hi.
Hey. How are you? Are you ready to pitch?
Yeah, good, definitely.
Okay,
I'm
perfect.
on a holiday, so I hope you don't hear the construction outside. But I'm on holiday.
Let's hope they're not gonna drill for 90 seconds. So I'm gonna add your video and remove myself, and the floor is yours to pitch.
Thank you. Did you know that without hemoglobin, our body would not be able to transport oxygen to our organs and we would probably die within minutes? Now, people with certain blood diseases can actually not produce enough hemoglobin, which, as you can imagine, causes a lot of problems, like severe pain, constant infections, or even death. One way to solve this problem is to switch on the production of so-called fetal hemoglobin. This is a special version of that normally stops being produced after birth by the child. In my PhD project, I'm working on a way to reactivate the production of fetal hemoglobin to create a cure for these severe diseases.
Based on Anna's explanation, I wanted to create an animation that represents the journey of the researcher and use the element of transformation. She is transforming the path the body takes to create these cells. So I wanted to incorporate that with flowing transitions and transforming shapes, like bubbles to cells or a face to a blood clot bomb. The viewer is guided through the health changes of the cells by use of color and and we encounter both diseases Anna's researching in some way. But in the end, they won't be sure whether or not they can cure the disease, or in this case, defuse the bomb, because we don't know the answer ourselves yet.
Yeah, I found it really cool to work together with an artist. I found it very inspiring, and it actually gave me a bit of a new perspective on my project. And I really like how Annalie brought this whole core concept of transformation into the art. So, yeah.
Thank you.
Thanks.
It is amazing, and I've seen you had the help of a cat as well.
Yes, we did.
Perfect. So
Very
well
essential.
done. Yeah, essential. Cats are always essential. Thank you very much. It's really pretty, and it was a pleasure to watch. Your links are in the chat, so if anyone wants to join you, to reach out to you to discuss your project, art or science and art, they can do it. Thank you very much for pitching. See you very soon on social
See you
media or in
soon.
real life. Bye.
Thanks.
Bye.
So now is the turn of Maxim Kolmatov and Aliona, sorry, that I'm going to bring here with me. Hello.
Hello.
How are you?
Yes, good.
Good? Okay, perfect.
You're hearing us all right?
Yes, I'm hearing you all right. It's all good. I'm going to put your slides and disappear. Unfortunately, the GIF doesn't really work on the platform, so
It's
we're going to
okay.
have to do with a still image, but you can describe and we can imagine what's happening.
Okay,
Let's
great.
go. Let's go, and I'm disappearing.
Okay, so you may remember from school that all of the blood cells in your body arise from special stem cells that live inside of the bone marrow. But what's been realized a lot more recently is that bone marrow consists of many different types of cells that are constantly interacting with each other, and then they can become friends or enemies, and sometimes they cannot live without each other. So the goal of my project is to try to analyze how these interactions between various types of cells can shape normal blood formation and different diseases. Yeah, okay.
Yeah, listening about Max's research, it struck me how his subject of research consists of so many similar yet very different small elements, and it reminded me of a herd of cats, which are also kind of similar, but each have their own character. And in this generative art project, I played with this idea of many similar elements.
Yeah, I was really impressed, and I found the art super interesting, and the idea of doing the actually animated version that with some simple aesthetics tries to confer these ideas of diversity and functionality between different cell types, and I'm really looking forward to seeing the final version.
Did you plan that apparition of the cat at the end? Because that was...
No, I'm sorry. You cannot control her.
Don't be sorry. That's perfectly on point. This is perfectly on point. You know, they are the inspiration of the art, so they might as well show up and say hi. Perfect. Thank you very much. Again, the links are in the chat if people want to reach out to you. It was really amazing to see your art. And normally it's a GIF, so we see the little eyes blinking, right?
Yep.
Okay.
Well,
but that's not. Yeah, it's not the final art. It's gonna evolve again, right?
A little bit, yeah.
Yep. Exactly.
Okay, perfect. So can't wait to follow that. Thank you very much to both of you. So we're going to say goodbye, and we're going to now welcome Gaurav. I don't really... okay. Okay, sorry, that was your version. Perfect. No. Yes, sorry. So we're going to now welcome Francesco Leonetti and Alessandro Ventura. Yes, perfect. Hello.
Hello. Hello.
How are you?
I'm good, thanks.
Thank you. So this is your slide. I'm going to disappear and you have 90 seconds to pitch.
Okay, thank you, Elodie. So hi everybody. So acute myeloid leukemia is a cancer of blood cells. In this disease, the classical therapy often fails. With this project, we aim to uncover the molecular basis of this phenomenon called drug resistance. And we started selecting several genetic elements, better known as enhancers, whose function basically is to boost the transcription, to identify and inhibit the proteins they interact with, and provide, in the end, a way to overcome the drug resistance using a combination therapy to definitely kill the cancer cells.
Yeah. So hi all. So this is my, okay, not really mine, but it's a collaboration of me with artificial intelligence. So with the guide of a human brain, my brain, artificial intelligence has been able to create the 3 images you can see. Basically, in order to create with AI, you have to describe as best as you can the picture you have in your mind, and then refine it with multiple parameters. So I've inserted keywords I've heard from Francesco, like DNA, tree of life, gene induction, and then I've added a personal touch, adding other keywords like blue sky, gold, landscape, and of course the favorite painter of Francesco is Salvador Dali. Thanks.
Thank you, Alessandro. I'm very satisfied with this project.
Thank you.
Well done. Thank you. Thank you for pitching it. It looks amazing. I love the diversity of all the art that we're seeing. It's really amazing. They're all reflecting the artist and all the different projects, because the ITN, it's lots of different projects, part of like the same big goal, but lots of different things looking at different biological questions. So it's really nice to see that translated into art. Well done, both of you. Thank you again.
Thank you, Elodie.
Bye.
Bye. So now we're going to welcome the next one. Yeah, perfect. We're going to welcome now Gaurav, Mandana, and Dimple again. Devadatta is not there today. So Gaurav, you have the big challenge of pitching on your own. Are you ready?
Yeah, more or less.
More or less. Perfect.
Okay.
I will take that as a yes. So you have 90 seconds.
Okay, thanks. So here's more or less my project. So enhancers are pieces of our genome that are important for making sure that the correct parts of the genome are used and in the correct proportion. However, enhancers themselves need to be used in the correct proportion. And there are certain roadblocks that are set in place to make sure that the enhancers are used in the correct way, which in turn ensues that the genome is read correctly and the cells can develop and live normally. The removal of these checkpoints can disturb the fine balance of the cell, not unlike how a malfunctioning traffic light could cause chaos on the roads. The runaway cars end up going down streets that they really shouldn't, which in this case could be enhancers of the streets. The ensuing chaos ends up killing the cell. So my work involves investigating the nature of these roadblocks that allow enhancers to work properly and support the healthy functioning of the cell. So how does this tie in with the art? The artist, Dimple Devdas, has illustrated this idea in this work of art, which depicts scales, which is supposed to represent how the genome is used judiciously. The clock may represent the evolutionary potential of the genome, where previously unused parts of the genome may hold the potential to become something more. And she couldn't be here today, but working with her was a very rewarding experience.
Thank you. Thank you very much.
Thank you.
Thank you for pitching. So again, we've put your links in the chat. So if anyone wants to reach out to Dimple Dyson there, or you that was there, they can ask more questions about the science part or the art part. Thank you very, very much.
Thank you. Bye-bye.
Bye. So we're gonna now welcome Natsuda and Natsuda. We're having 3 people, but only Natsuda is gonna pitch. So we have Natsuda, Kohamon, and Kiyachi. Hi.
Hi.
Sorry.
Kanyarat and Kochamon is fine.
And Kochamon, yes. Hi, Natsuda. How are you?
Hi, I'm good, thank you.
So I'm gonna put up your art, and I'm gonna remove myself, and you have 90 seconds to pitch. Are you ready?
Thank you. Yes, thank you, Elodie. I create computation tools and models to analyze and predict biological data. I am focusing on analyze the data from a technique that identifies a starting point of the RNA production in our cells, called CAGE. To complement CAGE, I am using another technique called ATAC that show the region in the DNA that are active in the cells. By combining them, I get more precise results and deeper understanding on the characteristics of the region I am interested in. However, sometimes the experiment cannot be carried out since they are time-consuming, difficult, and expensive. This makes it necessary to develop a computational model that can use what we learned from the previous experiment and trying to predict the active regions using only CAGE data. The research concept leads to the art project by my artists, Fang and Kate. They come up with the idea of this installation art, which composed of 2 layers. The main layer, which might look like a fuzzy, meaningless line, represents CAGE. However, when you layer it with the blue filter layer representing ATAC, you will see a beautiful drawing underneath. Moreover, if you don't have the blue ATAC layer, we expect that the computational model I have developed will be able to predict the beautiful picture, in other words, the meaningful active CAGE signal. What picture we will see is still a secret that we'll reveal in Marseille in October, as the artists want to surprise us all. Thank you.
What a teasing.
Thank you.
Thank you very much for your pitching. Now we have to follow what's going to happen in Marseille. And for those who are not there, all the links are in the chat, so you can follow everything that's happening from far away and everywhere in the world. Thank you very much, Natsuda. Well done.
Thank you.
So the next one to pitch, we're going to welcome now Olga Pushkarev and Anna Stankiewicz. I'm going to put them on. Give me a second. Sorry. Olga, hello.
Hi.
Hi, Olga.
Good, good. Thank you.
You're ready to pitch?
Yes, I am.
Perfect. And Anna is not there, right?
She's not there?
I don't see Anna. Sorry.
Okay. She was supposed to connect, but...
Okay. No, I don't see Anna. Let me just... Yep, I don't see Anna. Yeah, I see Anna. Okay. Perfect. Sorry. She's there. Hi, Anna. All good. Are you both ready to pitch?
Yes.
Okay. So I disappear and I'll let you do it. And give me a second to add your slides.
Perfect.
Yes.
Okay. Have you ever wondered why some people are more prone to certain diseases than others? While our environment and lifestyle matter, part of this differences comes from our DNA, making each one of us unique. And in my research, I'm trying to understand this incredible diversity we see in humans by looking at DNA Lego blocks that connect to each other and depend on one another. We call them chromatin modules. And by understanding how elements in this module synergize and which genes they involve, we can take one step forward towards personalized medicine.
And so the title of the artwork is Synergy. And this is a concept that I think reflects our collaboration of scientists and artists, as well as the processes of interaction and cooperation within these complex genetic systems. The artwork itself will be a representation of this, starting with a single image fragment that will slowly unfold like a map to reveal the larger interconnected shape. And the final ink drawing will explore complexity, connection, and this unique experience of trying to understand the inner workings of our genes.
I find the idea of unfolding like a map truly captivating because it provides a very powerful visual metaphor for the concept, and your work emphasizes details and really wonderfully highlights the complexity of gene regulation.
Yes, it was a nice challenge to figure out how to interpret the science, but also create interactive work that can be presented in both a physical and a digital format. So you can see now a little taste of what the final piece will look like in October.
Yeah, I'm really looking forward to see the final artwork and also the process of how you create it.
Thank you.
Yeah.
Amazing, amazing. Yeah, again, such a teaser to see the art in October. We're going to discover so many things. So again, you don't have to be there in October in Marseille necessarily. You might not be there in Marseille necessarily. So the links are in the chat if you want to connect with both of you. Thank you very much. It was amazing. Can't wait to see the final thing.
Thank you.
Well done. Bye-bye.
Bye. So now we're going to welcome Irina Ribeiro-Bass and Andres Mistrik. Hello, both of you. Hi.
Hi, Elodie.
Hi, are you ready to pitch?
Yes, we are.
Perfect. So you have a video. I'm going to remove myself from the stream. I'm going to put the video and you can start your pitch. Okay.
So our bones form a very special type of cells that have the ability to transform themselves into any cell type that exists in our blood. When we get ill, like for example, when we have inflammation, these cells can sense it. And then they start transforming into cell types that will defend us by fighting the cause of inflammation. However, there are some situations, like for example, when we get older, when this inflammation is chronic, persistent. These cells are constantly stimulated, and then they start losing their fitness or the abilities that makes them so special. This means that our lab is trying to understand how and why this is happening.
Andret, we don't hear you. Yeah. So sorry for the little technical issue. Do you have earphones or microphone? Something. Don't worry. We're gonna start. Sorry. Sorry, everyone. This is the technical issue you can get with
Yeah, a bit of technical issue, but I think we can solve it because we are actually very close from each other, just in different rooms.
Yes.
So we probably can make it together.
Okay, so I'm with you. Perfect. Yes.
Andra? Artist, come here. Make it from here, this computer. Okay, so
perfect.
Yes,
okay, so you know what? We're gonna start. We're gonna start from the start and pretend we haven't heard anything yet.
Okay.
Yeah, so
from the start, Andra.
Okay,
can you do it again from the start?
Okay.
Both of you. Let's pretend it didn't happen. Okay, sorry everyone watching us for the technical issue. So let's hear you pitch.
Now, so our bones form a very special type of cells that have the ability to transform themselves into any cell type existing in our body. So when we get ill, like for example, when we get inflammation, these cells can sense it, and then they start transforming into cell types that will defend us by fighting the cause of inflammation. However, there are some situations, like when people get older, for example, when the inflammation is chronic, persistent. These cells are then constantly stimulated, and then they start losing their fitness, meaning the abilities that makes them so valuable for us. In our lab, we are trying to understand why and how this is happening.
And so to illustrate Irina's project, I made a short visual novel which portrays this weird and complex world inside each and every one of us, telling the story of these cells battling chronic inflammation in a way as if it was happening on another planet. In this work, cells are impersonated as round creatures with eyes, and DNA mutations are shown as bugs, and they all kind of intuitively tell a story of this micro-universe. And so I hope it inspires whoever will see it to give a thought to the amazing worlds happening and hiding both around and inside of us, to maybe pay them some attention in their everyday life.
When I saw Ondra's animation, I thought it was amazing, and it was a great idea to portray the work that I explained to him. So I'm very happy to be seeing this in Marseille in October.
Thank you, Irina, and I'm really looking forward to present my work in Marseille. Yeah. And sorry for the technical issues.
No worries. Amazing. Thank you. Thank you very much, Ken, in October as well. Again, links are in the chat, so I hope people are going to contact you to ask you questions and to know more about the science and the arts as well.
Thank
well done, both of you. Thank you very much.
Thank you.
So, bye-bye. We're now going to welcome... suspense. We are now going to welcome Sarah Butt and Min Wang. So, hello, Sarah.
Hello.
And Min. Hello.
Hi. Can you hear me well?
Are you ready to... Yeah, we hear you well.
Perfect.
Are you ready? Are you ready to pitch?
Yes. Yes.
Okay, perfect. So I'm putting the video, I'm disappearing from the screen, and the floor is yours. Actually, it's going to be the other way around. Okay, see you in 90 seconds.
At the beginning of the pregnancy, many things could go wrong. One of them is called holoprosencephaly, or HP. The most visible sign is a narrow gap between the eyes, and my research focuses on understanding how the environment can influence it. Then, I explained the context to Min. She thought of robots.
Yeah, so this is what I think about this. Imagine a human is like a robot, and then a human brain is like a CPU of a robot, and the pregnancy is like robot cloning. And if a robot have an HPE malware, it may appear in close-up pair of camera eyes or have just one camera. During the cloning, the HPE malware could be copied to the clone. In ideal condition, the cloning can be right with low chance of activating the HPE malware. But if we ate the HPE malware with some booster like alcohol, cigarettes, or certain cosmetics, there's more chance of activating it, and we could get a clone with bad CPUs as a result.
Yes. Put simply, in human, if the gene causing HP is damaged, the brain can possibly develop wrong. But still, there are many unknowns. That is why, in the lab, we are trying to understand how these factors could increase the risks.
Sarah, this research is interesting, and I'm looking forward to more finding about how this disease appeared. I believe it will be very helpful for many people in reality. So thanks for your work.
Thanks to you too.
Well done. Well done, both of you. Thank you for the pitching. And again, we have the links that are in the chat. So if people want to connect with you and know more about everything you do, science-wise or art-wise, they can do it. Thank you very much for the pitching.
Thank you.
Bye. So now we are down to the last two ones. That went really fast. The last two pitching. So we are going to hear the one before last, Jin Wan and Ji Yung Feng are going to join us. So hello. Hello, both of you.
Hello. Hello, Ruby.
How are you? You ready to pitch?
Good, good. Yeah.
Yes, we are ready.
Perfect. So I'm gonna disappear and I'm putting your slides and you have 90 seconds. See you in 90 seconds.
Okay, so my research is to study what controls genes activity in the cells and how. So you can imagine a gene is a car driving on the road, and there is a power on button nearby the car, and we call it a promoter. It's a piece of DNA before genes to start the car. And there is a piece, a gas pedal, we call it enhancer. It's a piece of DNA far away genes to speed up. Now we found out power on button can also speed up, so we call it e-promoter. And so how is gene controlling e-promoter? You can see this 3D model to have an idea. So our science and art collaboration start from this model. So we can move to Ziju.
Yes. In my part, I'm inspired by Gene's DNA structure, especially the animation that he sent to me. Actually, I saw the repetitive gesture of twisting as an organic, mechanic, and generative body. This body can be considered as an extension of humans' hands, legs, or whatever. So I made a twisted structure by using flexible material, then combining the twisted structure of life conception and digital of the other digital elements at all. You can see the ray flowers on the right, like a digital flower made by super light clay and a table lamp, a keyboard made by baguette, and a radio made by fitness material. All of this conception and elements come to interact, and actually it's an installation art, and then to show the possibility of life in digital era, like kind of like this stuff.
Yeah, actually, this is my first time to collaborate with this artist, so it was a very nice and fun experience to discuss science and art with Zijun. So thank you very much, and we are really enjoyed.
Yes.
It is pretty well done. Thank you both of you for the pitching, and can't wait to see everything later. Really well done. It's amazing all your PhD's projects turned into art. It's really nice to see that. Especially it's not very common. It's really cool.
Okay,
well done. So again, the chats, the links are in the chats. So if you watch it on replay, you can still watch the live chats and get all the links to follow everyone. So if people want to reach out to you to talk about your art or your science or your project of science and art, they can do it. Thank you. Bye-bye.
Bye-bye.
So now we're having the two last ones to pitch for today. So it's going to be Artemis Zinglev and Ekaterina Onyufrenko that I'm going to call here. Hello.
Hey.
How are you?
Good. We are super excited to present our photo shoot.
So I just got the slides, so I'm putting them on. Give me a second. Sorry for the little the little technical thing. I'm importing your slides, and then you can present. Okay. Now it's there. Okay. Perfect. I'm disappearing, and whenever you need me to go next, just say next. Okay?
Yeah. Good. So hello, everyone. My name is Artemis, and today I would like to present our exciting photo shoot called Distance. Next, please. Yeah. So I'm quite interested in mutations like outside of genes in different regulatory elements, including enhancers. And these enhancer mutations, they are quite important in different diseases, including cancer and congenital diseases, as well as in predisposition events. However, these mutations are quite difficult to study, and to dissect these cases, usually we address 3D genome organization of DNA. Next.
The idea is that we put gene enhancer interactions into the context of human relationships. Since these interactions usually span significant distances, one can formulate the concept as, despite the distance in the metaphorical sense separating people, we can love and build relationships with each other. Our photo shoot features a guy, gene, and a girl enhancer holding some ornithorpe structure tied in the shape of heart. The composition of the frame is based on the reference from the previous slide. So the rope structure represents the DNA region between the gene and enhancer. Next.
Yeah, so generally we organized this photo shoot and involved lots of people. I think Russia is kind of popular now for the mixed media skills, so we'll add some of this stuff to the end version before the event in Marseille. We'll also add some really nice frame showing like some DNA, 3D genome organization, and all the stuff. Follow us on Instagram to see our next projects and final results of this photo shoot, and see you in Marseille.
Thank you. Thank you very much. So yes, the links are sent in the chats if you want to follow you and know more about the science and the art project. Thank you very much. Thank you. Bye-bye. Perfect. So we are at the end of this pitching event. Thank you very much for following everything. So you've heard about lots of different projects. You have all the links in the chats. You can follow, obviously, each of them, but you have the Enhpathy consortium that you can follow. You have the website, you have Instagram accounts, a Twitter page, and a LinkedIn page as well. So please, if you want to know everything that's happening in the science side and about this project, follow them and follow individual ones as well. It was really nice to have you here for this live, and that's it. So I'm gonna close the live and say thank you again for being there. Well done, everyone. It wasn't a very easy task, but they all have done very, very well. Bye, everyone.